Professor Rita Alloway presents her research into the safety and efficacy of generic transplant medications. Her practice areas include kidney, liver, and pancreas solid organ transplantation. Her work in transplantation pharmacotherapy has generated numerous publications. Professor Alloway's research efforts focus on the goal of individualising immunosuppressive regimes in the transplant patient. The objective of this video is to provide an overview of the drug approval process and its applicability to immunosuppressants used in solid organ transplants.
Hello, my name is Rita Alloway, Research Professor in Internal Medicine in the Department of Internal Medicine, Division of Nephrology at the University of Cincinnati. I serve as the Director of Clinical Transplant Research and have done significant work in the comparison of pharmacokinetics of immunosuppressive agents. Today, we're going to focus on generics, a good medicine or cheap fix in our kidney transplant recipients. In terms of disclosures, I have financial relationships within the past 12 months with a variety of transplant pharmaceutical companies who are studying immunosuppressants. I serve as on the advisory board and speakers bureau for Veloxis, Sanofi, and I am also provided an honoraria today by PharmaCorp to present this talk. This presentation does not include discussion of off-label or investigational drugs. The objective of today's talk is to identify the pharmacokinetic parameters which are evaluated to determine bioequivalence in healthy volunteers, review the current bioequivalence data with generic immunosuppressants in kidney transplant recipients, and identify the impact the adoption of generic tacrolimus has had on U.S. transplant recipients. Now, before we dive into the topic of generics, it's important for you to understand the definition of bioequivalence before you can make the step or the jump to their effectiveness. So this is a very regulatory intense definition, but let me go through it for the sake of the topic. So the regulatory definition of bioequivalence is the absence of a significant difference in the rate and extent to which an active ingredient or active moiety in pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed studies. Okay, so let's translate this to transplantation. So what is the absence of significant difference? Okay, there are bioequivalence parameters that we'll discuss more as we go through this topic that focus on what is the significant difference that would determine a difference in our patient outcomes. Also, we want to measure the rate and extent of which tacrolimus, in this case, reaches the bloodstream. So, and we'll talk about what pharmacokinetic measures we use to quantitate rate and extent. Then we move on to the active ingredient. Again, in this situation is tacrolimus. And the pharmaceutical alternatives are just the variety of formulations that we have within transplantation. And you have to make the jump that if the same amount of the active ingredient is in the bloodstream at the same rate and extent, that it would exert the same mechanism of action at those sites of where it is exerting its pharmacokinetic effect. So when we get to appropriately designed studies, there's a lot of controversy surrounding this as well. So the standard design of a bioequivalence study is very simple, actually. It's a two-way crossover study in 24 to 36 healthy adults given a single oral dose of the generic or the brand product, and the pharmacokinetic parameters are calculated from the time concentration curves that result from these blood samplings. The pharmacokinetic parameters, as you see here, again, 24 to 36 healthy adults are randomized to see just a single dose of either the brand or the generic product, and then a variety of blood samples at very intense time points are collected. When the tacrolimus levels are run based upon those time products, this pharmacokinetic curve here is constructed. After a significant washout period, the exact same healthy volunteer is given the alternative product in a single dose, and then again, this pharmacokinetic curve is constructed for the corresponding product. And from this curve, you see that the time in hours is on the x-axis, and the concentration, for this case, tacrolimus, is on the y-axis. So when we begin to assess the rate and the extent of the exposure of these patients, we begin to look at the pharmacokinetic parameters of AUC and Cmax. Cmax here, as you know, is the peak concentration that occurs after an oral dose. So this is measuring the rate of exposure, and the area under the curve is just a calculation of this area, and it represents the extent of which the exposure of a generic product is available within the circulation. FDA bioequivalence standards focus on two one-sided test procedures that are also called the 90% confidence interval approach. Okay, this recognizes that there will be differences in the mean values between the two formulation treatments. However, it provides reasonable assurance that the mean treatment differences are acceptable. The statistical approaches and guidance recommends that confidence intervals between 80 to 125% for AUC and Cmax of long transform data would provide you the assurance that there are no significant differences that will result in treatment differences. Now, one of the most common misunderstandings is when people look at the 80 to 125% range, they think that there's potentially a 45% shift or potential difference in these products in a patient. However, that's just not how confidence interval testing works. When you incorporate the variability of these products into these calculations, it really makes the mean ratios very, very different and a lot smaller than this 45%. And I'll show you examples of that. In Europe and actually in the United States, there have been special provisions made for narrow therapeutic drugs such as tacrolimus that actually keep the Cmax the same at the 80 to 125%. However, they narrow the area under the curve confidence interval testing to 90 to 111. And we can talk about more if we have questions regarding this. So in the United States, what were the public concerns regarding immunosuppressants? And to be honest, the concerns that have happened in the United States are no different than have happened in other places in the world. And to take it a step further, the concerns related to tacrolimus and transplantation are no different when their generic alternatives for cornerstone agents and treatments in other therapeutic areas become available. So there is concern because basically most of us as providers really don't fully understand this process. And so it takes some time to understand it. So what were the public concerns? The public concerns were that standard bioequivalence approaches based upon two-way crossover studies in healthy individuals and comparison of the pharmacokinetics of brand to generic products based upon average bioequivalence is not a valid approach for the approval of generic immunosuppressants in transplant patients. So the focus was on the lack of agreeing with the design of the study, the use of it in healthy volunteers and not transplant patients, and the comparisons of generic to brand. And the lack of data in transplant recipients, especially in high-risk transplant recipients, which are defined most frequently in transplantation as poor absorbers or patients that express cytochrome P450A genotypes. And the need to recognize that tacrolimus is a narrow therapeutic index drug, therefore, it may need more strict bioequivalence standards applied to this. And also the lack of comparison of data between bioequivalence among not only the brand product, but the other generics that the patients may be switched between. So here for your reference, there are several consensus documents that were published on this in the US and Europe and Canada, which highlight these concerns. So, with these concerns in mind, we began to explore the science between bioequivalence testing. And what is very interesting is that bioequivalence testing, when it is discussed in the relevance of generic products, appears to be a dirty word, or it's associated with something that's an ineffective process and it's not scientifically balanced. However, bioequivalence testing is used throughout pharmaceutical industries to provide us new formulations quite frequently. Even when there are changes in the manufacturing process that could result in changes in bioavailability of a product, the manufacturers have to repeat bioequivalence testing in healthy volunteers to show that their manufacturing process did not change the final product that they are providing for patients to consume. So when you think back just specifically in transplantation at the products that were actually used in the phase 3 studies to get the product available on the market. For cyclosporin, it was Sandimmune oral solution. For mycophenolate, it was CellCept 250 milligrams. For sirolimus, it was Rapamune oral solution. And for tacrolimus, it was Prograf 1 milligram capsules. However, if we look into transplantation, we all know that we have a variety of different formulations. How did these formulations become available and how did we begin to put trust in them? Okay. The products became available through bioequivalence testing in healthy volunteers that showed a Sandimmune capsule was bioequivalent to the oral solution, or a Neoral capsule was bioequivalent to Neoral solution, or a 500 milligram CellCept tablet was equivalent to a 250 milligram CellCept capsules. And so on, specifically with Prograf, when we went to 1 milligram capsules to 5 milligram capsules, and now with the granules. So bioequivalence testing is used within the industries a significant amount, and not just for generic drug approval. Therefore, the process has been rigorously tested over and over again. So, in brief, I will give you just an immunosuppressant generic summary of what occurred in the US. In 1998, there was the first modified release cyclosporin liquid that was marketed, but it was withdrawn from the marketplace because it was not bioequivalent in apple juice. It was bioequivalent in milk, water, soda, chocolate milk, etc. But when they did the test with it in apple juice, it failed. Therefore, it had to be withdrawn from the market. In 1999, the first modified cyclosporin tablet generic was marketed. And actually, what was really the game changer was in 2009, both the first mycophenolate mofetil generic and the first tacrolimus generic was marketed. And now we have, almost 15 years of experience with these agents here in the US. However, if you look in 2013, you can see that there was significant generic market penetration of these products in a relatively short period of time. And I'll give you more data on this at the end. A few years later, in 2014 and 2019, generic sirolimus and everolimus, respectively, was introduced to the market within the United States. So if we go back and you think about the public concerns that were raised in the United States, study design was a big concern. And at that time, the FDA or the NIH submitted a U01 RFA, request for application, to study generic tacrolimus in transplant recipients. And so me and my team submitted a study for this, and we were awarded funding for this. However, before there was agreement to proceed with this study, we met with not only the FDA, but also simultaneously the American Society of Transplantation and the American Society of Transplant Surgeons, who were able to provide input on this study before it was initiated and really provide the opportunity to give input into the result. So the design of this study was that it was a fully replicated, partially blinded, 6-period crossover study in 2 centers. Our population was the typical transplant population, adult patients that were greater than 6 months post-kidney transplant, and they were on a stable twice-daily dose of either Prograf or generic tacrolimus, and they were not receiving any other drugs that interfere with tacrolimus metabolism and pharmacokinetics. Our intervention was that upon evaluation of the healthy volunteer data, the bioequivalence healthy volunteer data for all the generic products, we would attempt to predict what would be the 2 most disparate tacrolimus generic products. And from that, we identified what would be a potentially generic high exposure, it would give the highest concentration based upon the healthy volunteer data, or generic low, who would potentially be the lowest concentrations in potentially transplant patients. So with this design, we had 6 pharmacokinetic assessments that were done over a 12-hour period where we gave brand twice, generic high twice, and generic low twice. And the beauty of this design is that we are able to compare brand to itself, brand to generic high, brand to generic low, and also generic high to generic low. Now, the reason it was important for us to compare brand to itself is we as providers need to recognize that even when we have a patient on immediate release brand product and they are taking it continuously, when they come back to clinic and they have their levels drawn, they still have variability in their levels, even though they've been on brand product all this time. The occasional patient still has a rejection or complications, even when they've been on the brand product. So we have to appreciate and accept that those potential complications occur with this type of product, and we have to be able to differentiate, is it a patient effect or is it a formulation effect? And that's what this study attempts to do. So our comparator groups, as we talked about, looked at generic high versus generic low versus Prograf, and then we compared the 2 generics to each other. And our outcome of interest were tacrolimus product within subject variability, our average and scaled average bioequivalence testing, and observed if there were any formulation effect of tacrolimus variability and exposure. So this is our design sequence. Again, we only conducted this in 36 transplant patients. And as you know, unlike healthy volunteers, they had to stay on their immunosuppressants for the entire time. We couldn't take them off their immunosuppressants. Therefore, they maintained on a generic formulation the entire time. We measured not only the tacrolimus levels, but also the metabolites. And we did genetic phenotyping on all of our studies. So all our study subjects. So if you go down to the randomization sequence at the bottom, you'll see that patients were screened on day -14, and they were brought in for randomization on day 1. And on day 1, they were assigned to either sequence 1, sequence 2, or sequence 3. And from that day, they were put on the exact same dose of the formulation that were within their sequence. So either generic high, generic low, or brand. And they were given this product for an entire week, and we monitored their drugs adherence with a MEMS cap technology, which let us know when they took the cap on and off the bottle. And we did variety of level, trough level assessments during this time as well. However, on day 7, they came in for a full 12-hour PK assessment. After that PK assessment, they were converted on the exact same dose to the next product within their sequence. And then again, 7 days later, came back for a full 12-hour PK. So you can see this occurred until 6 full 12-hour PKs were conducted with the patients receiving each of the product twice to assess for variability. So in terms of blinding, There was discussion by our teams for over encapsulating the products so that the patient nor we could visually be able to compare what formulation they were giving. But there's concern that over encapsulation can impact dissolution and subsequently the pharmacokinetic parameters. So it was determined not to do that. However, we attempted to assess alternative methods of blinding. So in terms of the PK study site, the subjects were randomly assigned to study drug sequence by the IDS pharmacy. And yes, when they came in, we could see which product they were randomized to, but we didn't know the sequence. However, when we at the site drew the drug levels, we blindly labeled them with only patient ID and time of the samples, and these samples were sent to the analytical site that was independent from us, and they ran the levels and were be able to provide us the individual level results. There were two additional analysis sites. One was our a pharmacokinetic analysis site, which took these levels and without knowing the sequence or the product that the patients were on, they evaluated the overall AUC, concentration max, time to max, and Cmax for each of those pharmacokinetic time periods. And then those ultimate results were given to an independent statistical team to assess for bioequivalence. And I will go ahead and keep the audience blinded to formulation until the conclusion so you can have a feel of the blinded results. So the demographics for our kidney population was a mean age of 52, predominantly male, only 17% African American race. The transplant donor type was equally divided between deceased, living related, and living unrelated. The time post-transplant was approximately 4 years. Over a third of the patients had diabetes, and the maintenance immunosuppression included 14% receiving steroids, 100% receiving mycophenolic acid, with a mean tacrolimus daily dose of 5 milligrams a day. So I'm going to take a bit of time to orient you to this slide. These are the pharmacokinetic results that were produced from our study. Again, I want to orient you to time over the x-axis and these tacrolimus concentrations on the y-axis. And each of these open circles here represent a time of which we drew a tacrolimus blood level in that patient after they had taken their dose. So this is orienting you to the time we gave product one, the first exposure, and you can see the values here, okay? The first thing you may say is, okay, why are the standard error of the means so high for these lines here? And if you remember, these are transplant patients. We have to give them the exact same dose that they're on to maintain their target. So this is not a dose that's the same between all groups. It's not a dose that's adjusted for milligram per kilogram. This is the dose in that individual patient that was required to get the optimal trough. And you can see this is the variability that results. Okay, so I began to lay over these plots, and this you'll see the first product, the second exposure. This you see product two, first exposure in green. Product two, second exposure in green. Product three, first exposure in blue. And product three, second exposure in blue. And when you look at all of these curves laying on top of each other, there were no statistically significant differences at any time point here. And when you look at the formulation, even during the most variable time during the peak, we're really only separated by a few nanograms per ml dosing. And again, I want to point out that these were the doses that were individual for each patient. So from these pharmacokinetic curves, we were able to calculate these pharmacokinetic metrics. So the AUC, the Cmax, the Cmin or trough, and the time to maximum concentration. And when you looked at product one, product 2 and product 3, and you compared their geometric mean and their 95% confidence interval, you can see that they were very similar across the board. When you look at Cmax, again, which is going to be the most variable, you can see that they are very similar as well. The concentration min, or the trough, you'll see is very similar. And the time to maximum concentration is very similar within these three products. Now, there is some debate, as I mentioned before, as for therapeutic drugs such as tacrolimus, should we have more strict bioequivalence standard testing? And I mentioned to you before, in Europe, they have chosen to narrow the area under the curve for tacrolimus to 90 to 111%. In the United States, they have offered an alternative testing process for therapeutic index drugs, which is called scaled average bioequivalence. And actually what this does is it takes the within subject variability of the reference product, and based upon the lower the within subject variability is, it begins to narrow the bioequivalence limits. So basically, the bioequivalence limits can never go over 80 to 125, as you see here, despite the high variability of the product. But if a product shows that it's limited variability, it will narrow. So it will be more difficult for them to be within the the bioequivalence standard testing. Now, I've published significant numbers of papers on tacrolimus, and I have talked about it being a highly variable drug. And that is true whenever all sources of variability are taken into account. However, when you begin to minimize those sources of variability, like being very strict on the time of which the patient takes the drug, controlling the food that the patients eat as it relates to the percentage of fat in their diet, and being very strict on the dose that is given and following adherence, it's going to be interesting. You're going to be surprised the within sense during a well-controlled study. So again, that was one of the advantages of doing this study and being able to compare the products to each other. When you look at tacrolimus area under the curve, the reference within subject variability of product 1 for AUC was only 12%, which shows you that, again, when we have patients that are very consistent and the patients are very adherent with their diet and other things, this product is really not a highly variable drug. Even when you look at product number 2, the generic, and you compared it to itself, the within subject variability was 14.6%, and then within product 3, it was 15.8%. But in scaled average bioequivalence testing, this reference within subject variability is very, very important to calculating the subsequent scaled average bioequivalence limits. Another point I want to make before I move on in this slide is the AUC geometric mean ratio. And as we talked about before, think the concept of there being a potential 45% swing in AUC with bioequivalence testing is just not true. When we compared these to each other, there was only a 5% difference when comparing the two products for AUC, almost spot on 100% 0.02 ratio when product 2 and product 3, and 99.98 when compared product 3 to product 2, or just the inverse. But again, based upon within subject variability, based upon this percentage, a scaled average bioequivalence limit was assigned. And so with a product that has low within subject variability, as we said before, the bioequivalence limit testing was narrowed to less than 80 to 125%. And these were what would be defined for us to meet it. And then the next question becomes is, okay, these are the scaled average bioequivalence limits that you need to meet bioequivalence. What were the actual observed bioequivalence intervals? So these are the observed bioequivalence intervals. You can see that all of these fall within the 80 to 125% that you would look for average bioequivalence. But even the more stringent standard of scaled average bioequivalence, you can see that these smaller, more narrow ranges fall within the observed 90% confidence interval falls within the scaled average bioequivalence. So this is what we observed, and this is what we calculated. And the observed 90% confidence interval falls within the scaled average bioequivalence. So the results of this is that it would pass FDA average bioequivalence, FDA scaled average bioequivalence. And when you actually look at the 90 to 111.11% of the EMA bioequivalence testing, it would actually pass the EMA bioequivalence testing for AUC as well. Just moving on to Cmax, I'm not going to go through and describe this all again, but you can see that yes, the within subject variability is slightly higher for the Cmax pharmacokinetic variable, as you would expect. The scaled average bioequivalence maintain the same, 80 to 125. And again, the observed bioequivalence estimates fall within that and they pass our standards. Even though Cmin is not a bioequivalence testing parameter for bioequivalence testing, it's a very important pharmacokinetic parameter to our transplant community because it's what we use to monitor the patients and also needs to be a good reflection of the overall AUC. And as you can see here, we calculated the scaled average bioequivalence, and our observed bioequivalence standards actually fell within the scaled minimums or limits, and therefore it passed FDA standards for average, scaled, and it would have passed EMA more narrow standards for this, even though the EMA only applies to the AUC. Now, granted, this was a short duration study, and we didn't expect to see any acute rejection, changes in graft survival or patient survival, and we didn't see any differences there. There was one serious adverse event of pyelonephritis that resulted prior to study drug administration. However, as with many of our transplant patients, 71% of a kidney patient population reported some type of adverse event during this time. And these are the way that they panned out as it relates to products, product 1, product 2, and product 3. And there were really no differences that we saw in the adverse events by formulation. So in conclusion, this study was whose design received substantial input from the FDA, the American Standard and generic bioequivalence based on both conventional, average, and reference scaled average bioequivalence in stable kidney transplant recipients with no difference in AUC, Cmax, or the trough. There was comparable within subject variability that was observed for all of our formulation. There were no observed changes in the organ function with similar adverse events. And although the data wasn't presented, the covariants which affect pharmacokinetic PK parameters of tacrolimus, such as gender, hematocrit, African American race, or CYP3A5 expression genotype, did not impact the pharmacokinetic parameters. So to unblind the audience now, and you go through product selection, you can see that product 1 was our brand product, product 2 was what we predicted to be generic high, and product 3 would be what we predicted to be generic low. And the product selection that we had, and these were based upon the products that were available at the time we did the study in the US. There has since been more products available. Brand was obviously Prograf. Our generic high was Sandoz Pharmaceuticals, and what we predicted from Healthy Volunteer to be our generic low was Dr. Reddy's formulation. And from the observed pharmacokinetic parameters, there were no statistically significant differences in any of the pharmacokinetic parameters by formulation in transplant recipients. So the next question you may want to raise is, okay, how do you compare the products that you tested in your study with the Pharmacore Tacrolimus generic? And the only thing that we can really compare are the generic testings that were conducted in healthy volunteers. And so when you look at the generic testing for tacrolimus, the 5 milligram capsule under fasting conditions, of note, they also do them under fed conditions as well. But in fasting conditions, you can see with Sandoz, these were the point estimates that were observed when Sandoz generic was compared to brand in healthy volunteers. When Dr. Reddy's product was compared to brand in healthy volunteers, you can see these were the point estimates that we observed. And when you look at Pharmacore compared to brand in healthy volunteers, again, these are the observed point estimates that we see when we're looking at the pharmacokinetic parameters for Pharmacore generic. Just for completeness sake, I do want to point out that there is a systemic review and a meta-analysis of biopsy-proven acute rejection and bioequivalence that's available in the literature. It was published in Transplant International in 2020. And again, they found no difference. If anything, there was some evidence that suggested that there was lower biopsy-proven acute rejection with generic tacrolimus looking into de novo use. But this is available if you would wish to look at it. So the real question becomes is, okay, what happens when you use generic tacrolimus or generic MMF for that matter in a significant number of transplant recipients? So in the United States and in other parts of the world, there have been a significant uptake of generic product within the transplant community. And if you look at generic tacrolimus, these dashed lines that you see on these graphs are when a new generic tacrolimus formulation was introduced. This is focusing on tacrolimus, and this is focusing on MMF. And again, you can see here what's different from MMF is that there were already 4 MMF products that were FDA approved before they were even marketed. And when they were marketed, you can see that there was a very, very quick, like within months, conversion to where half the patients, these lines transversed, and basically half the patients were receiving generic and half the patients were receiving brand. Now to current, well over 80 to 85% of patients in the US are receiving generic MMF. And I want to point out, similarly with tacrolimus, there was initially only one branded product that was available. One generic product that was available. And when it was introduced, there was a relatively quick, not as quick as MMF, but a relatively quick conversion to 50% of the patients on the market receiving generic. But as different and additional generic products were introduced, you can see there was a higher market penetration of generics to where 75 to 80% of patients in the US are taking generic. Now, the other point that I want to point out to you is that we in the US, we were converting to generic tacrolimus and generic MMF, as you can see right here, at the exact same time. And we did not see any dramatic changes in our patient outcomes during this time. If anything, our outcomes continue to improve as it relates to biopsy-proven acute rejection and patient and graft survival during this time. And there's data that show that despite such a large percentage of our marketplace receiving not only generic tacrolimus, but generic MMF as well. And again, this is just another graphic of something similar, showing the percentage of patients that were dispensed a generic immunosuppressant during this time, where you can see that most patients were prescribed a generic immunosuppressant at some time during this assessment of these data. So it's important to understand what the cost of transplant immunosuppressant cost advantages, if they exist. So in the US, we have really a 2 separate payer model, where one is a government model where they have Medicare, And then other is private insurance, where the patients pay a percentage of out of pocket. Even in our government funded system, 80% of their immunosuppressants are covered with 20% out of pocket. There have been some recent legislation that this has changed, so hopefully this may go away. But prior to this, this was an analysis of the Scientific Registry of Transplant Recipients, looking at kidney and liver transplant recipients who had received Medicare Part D prescription drug benefits or had received private pay. And with the induction of tacrolimus generic, there was a decrease in the cost of the drugs by up to 50% for Medicare Part D payments. And it resulted in an annual cost saving when the patients had to pay, 20% of the difference, up to $2,700 per patient per year of those that actually had Medicare. And when you look at CellCept, again, the percentages were slightly higher, with a cost reduction to the government of 55 to 67%, and then the cost of the 20% that the patients were having to pay annualized to 2,500 to 4,200 over the year. When you look at patients that were paying out of pocket and had private insurance, you can see that the percentage decreases were actually greater for what the drugs were being paid for by the provider when tacrolimus and CellCept was used, but the actual out-of-pocket expenses to the patients were less, but still decreased a significant amount over a year for those that had private insurance. So with that, I hope I've been able to describe to you not only the science behind the bioequivalence testing for generics, but also the clinical impact of that as well. And with that, I'd like to thank you for your attention and also open the floor to any potential questions. Thank you.
Australian generic Tacrolimus market share currently sits at less than 10%. If uptake increased to 70%, there's an opportunity for the PBS to save 10 million Australian dollars. This cost saving could be reinvested into discovering new medicines. When considering generic medicines, it's important to remember bioequivalence testing is used throughout pharmaceutical industries to provide us with new formulations. Without bioequivalence testing, innovators wouldn't have released new formulations, including Prograf 5mg. Generic and branded Tacrolimus passed both FDA and narrower EMA standards during testing, with no notable differences in patient outcomes.